Hello. Pull. Oh. Woo. Yeah. Woohoo.
Assalamualaikum everyone. My name is Nur Inshira binti Nurman for practical 1, which is basic concept of statistics and variation. This experiment has been conducted by using 3 sets of leaf. There are 3 leaf type of variation being measured, which is our length, width and height. The objective of this experiment are, firstly, to calculate and describe the following description statistic: mean, variance and standard variation. Second, to understand the concept of correlation. Lastly, to determine the relationship between the 2 variations.
Hello everyone, my name is Nur Emily Sofia, and in this video, we will explain a discussion and conclusion regarding the Lab 1 and Lab 2. So, for the first part, I will explain about discussion number 1 and 2 of the Lab 1. First of all, what is differences or similarity of the mean of length between different sets of leaves? So, this experiment involved measuring length, width and height of 3 sets of leaves. Descriptive statistics were calculated and correlations were studied between length and width. The mean length of the leaves was 11.22, 9.51 and 12.68, with differences due to errors during collection. The upper part of the plant had smaller leaves due to increased water loss, while the bottom part had larger ones. The leaf size also influenced by age and genetic factors. The mean width was slightly different, with differences due to genetic and environmental factors. Precautions included collecting leaves from the same part of the plant, using correct significant figures and using high accuracy apparatus. Moving on to the second discussion, which is what is meant by variance. The leaves have different measurements of width and length. What is the meaning of this statement, and what is meant by standard deviation? So, variance in mathematics is the difference between the sum of square observation and the total observation squared per total observation. It measures how far a set of numbers is spread out from their average value. In an experiment, 10 leaves for each 3 sets with 30 leaves in total resulted in different measurements of width and length. The variance in length and width for each set was 13.3822 and 6.61, 14.016 and 2.8262 respectively. This information is crucial for visualization and understanding the data being considered. The square root of variance is used to calculate the standard deviation, which measures the variation of dispersion of a set of values and how measurements are spread out from the average or mean. Standard deviation can be divided into low and high standard deviations, with low standard deviations indicating closeness to the mean and high standard deviations indicating a wider range of values.
Question number 3. Why is it necessary to have different set of measurement for the measurement of leaf width and length? Having different set of measurement for the width and length of the leaf is useful in data because it allows for comparison between different set of leaf and to ensure a consistent result. Is it necessary to make assumption about the distribution of the leaf and the relationship between width and length? By having different set, we can compare our result with the other set. This is done to ensure that the experiment produces an accurate and consistent result. Conclusion of the experiment highlight the key concept of statistics and variation. Descriptive statistics allow us to summarize and present the data effectively, measure of central tendency such as the mean, median and mode, provided insight into the typical or average values within the data set, measure of dispersion such as the range, variance and standard deviation, help us understand the extent of variation present in the data. In conclusion, this experiment aims to introduce and explore basic concept of statistics and variation. Understanding variation and statistics enhance our ability to interpret and utilize the data effectively.
Hello again! For experiment 2, which is factors that affect the distribution and abundance of organisms. In this practical, we will expose several quadrants of 5 meter x 5 meter at each forest type. Students also need to observe the abiotic factor of a study site and classify the species of plant in that site. The objective of this experiment is to, firstly, to introduce the simple concept of quadrant. Next, to enhance the power of observation. Moreover, to record the difference in plant distribution within the quadrant. Lastly, to infer from nature observation the possible factor that affect the distribution and abundance of organisms.
Assalamualaikum and hi, my name is Norini binti Abdullah. Today I would like to present the discussion of post-lab practical 2, which is sampling technique. I will talk about question number 1, 2 and 3. Question number 1, define the term quadrat in practice. The answer is, quadrat is some random locations where in practice should plot the area that has been chosen with quadrat that has been set, which is by 5 meter times 5 meter a specific area. In addition, within the quadrat should all trees and several types of organisms to make observed species name, the total number of organisms that has found in the area. The results of different species in quadrat must be recorded in data to compare between each type of tree that has been chosen within the entire habitat. The method depends on linear measurements between random points and adjacent individuals and between adjacent pairs of individuals. A quadrat is used in practice to mark out a specific area of the plant communities to be sampled. Sampling unit of area was taken in the area of definite size. The quadrat sample must be taken to know about the species that are occurring in this practice. Question number 2. What are the two basic requirements of all the quadrat techniques? Okay, number 1. The area of the quadrat counted be known so that the density can be calculated directly. Next, the organisms must remain motionless during the counting period to ensure that they are counted and do not go missing. Question number 3. What would be the advantages and disadvantages of using a larger or smaller quadrat to survey the sedentary organism? The advantages are that it is inexpensive because few materials are required. It is also a feasible and simple method to use when compared to other sampling methods because quadrat plots are uniform in size and shape and distributed randomly throughout the sample area, making the study design simple. Next, the disadvantages are larger quadrats can provide more accurate estimates of overall densities, but they are more difficult to implement due to the time required to place and sample them. Not only that, study errors also may occur. Errors frequently occur when quadrats are too large, too small, or improperly spaced. Aside from that, inconsistencies in counting and omitting species that are partially within boundaries can occur.
Assalamualaikum and a very good day. My name is Muhammad Shahrizlan bin Mustafa Bakri and I will be presenting about question 4, 5 and the conclusion. So let's start with question number 4. Explain the issues concerned by ecologists in conducting the sampling method. The sampling method is relatively simple, involving simply by counting the number of organisms of interest in a defined area. However, there is a sampling issue that can be divided into several issues. First, consider the sampling method that can be used to collect the necessary data. Second, methods for selecting sampling locations to limit potential bias and obtain the best estimates of target parameters. Finally, methods for reducing the number of samples taken while still providing an accurate estimate of parameters are discussed. As a result, ecologists have developed several basic sampling approaches to deal with sedentary organisms, the most common of which is plot sampling. Moving on to question 5. Discuss the factors that would have caused the difference in the number of plant species in quadrat. The quadrat sampling technique was used in the experiment to identify and measure the plant distribution in a specific area. All plant species were observed and documented, and their distribution in the area was estimated. Based on the results, it is found that the Cladnopteris linearis sp dominated both areas Site 2 and Site 3 observed. It is one of the most widely distributed ferns of the wet Old World tropics and adjacent regions, including Polynesia and the Pacific. This plant is well adapted to tropical and warm subtropical environments, which is very suitable for Malaysia's climate. Several precautions were taken to reduce the number of errors and obtain accurate results, such as ensuring that the strings of the quadrats were tight to obtain exact squares. This ensures that all species in the quadrat can be precisely counted. Except from that, the quadrat is placed in an area with a wide variety of plants in order to obtain as many results as possible and to easily identify species that dominate the area. Moving on to conclusion. The objective of this experiment is to use the simple concept of quadrat to record the difference in plant distribution within the quadrat while observing the factors that affect the distribution of the plant and at the same time enhancing the power of observation. Through observation, it was found out that the factors include but are not restricted to the sizes of plants and the types of soils. The data was then used to predict the population of the species found within the environment rather than a small area. However, external factors such as animals and weather can affect the plant population, thus affecting the data.
So, let us move on from our experiment and look at something we are all familiar with. That is our community ecology. My name is Nur Inshira binti Nurman, and I would like to inform you about community ecology. We left at 8 AM for Taman Rimba Lenggeng in Lenggeng, Negeri Sembilan for our field trip assignment. We were briefly joined there by our lecturer and a local. Tourist soldiers and researchers are frequently on the area. Following that, we were divided into lab teams and began our community assignment. The goals is to look around and select from among the many creatures present, then apply what we have learned about community ecology to the creatures' interaction with their surrounding and the other species. What exactly is community? A community is a group of population of various species that live in the same location at the same time. When the population of zebras, bluebirds, deer, lions, monkeys, meerkats, mushrooms, trees, and grasses all live and interacted in the same place at the same time, a community is formed. Based on our research, we can use 4 community ecology concepts in our video: interaction, competition, predation, and prey defenses. To begin, interaction occurs when a single species population interact with other species, and this interaction can be: first, both or one party gains a positive benefit, also known as mutualism and commensalism, which is one organism benefit while the other is unaffected. Second, negative is harmful to both. Some of it is due to the competition. Third, a neutral party benefit. Predation, herbivory, and parasitism are examples. So let's go deeper to learn everything there is to know about our community ecology. This interaction can then be classified as a competition. 2 species use the same limited resources. This interaction is harmful to both negative entities. There are 3 types of competition. The first one is resources or exploitative competition. This occurs when one species prevents another species from accessing a resources simply by consuming it first. For example, consider the competition for light amongst the trees in a rainforest. As seen in the video on the left, the older taller trees effectively create a canopy absorbing the available light. As a result, the young small trees lack access to resources and are less likely to survive. Another type of competition that we can include is competition between birds for berries in cardinals. Second, interference competition. One species actively suppresses the foraging, survival, or reproduction of another, whether chemically or behaviorally. Interference competition does not necessitate a direct battle for resources. As an example, interference competition between colonies is common in ants, where it can prevent competitor from physically accessing a resources, either directly by fighting or indirectly by segregating the colony foraging areas. Third, diffuse competition. It has more than one competitor. Diffuse competition, rather than a single strong competitor, can exclude a species or greatly reduce its number through competitive interaction with a specific combination of other species. Diffuse competition implies that native species in community compete with one another for resources. For example, among plant, reptile, birds, and mammal species. Furthermore, predation, which is one species eating all or part of a second species, neutral. It includes the exchange of energy and nutrient within a community. Herbivory, carnivory, and parasitism are some examples of predation found in Taman Rimba Lenggeng. First and foremost, herbivore. Herbivores are animals that eat green plants or their seed and fruits. Plants are typically harmed but not killed. One animal consumes a portion or entirely of plant species, neutral. Small fish and zooplankton, for example, consume algae and phytoplankton. These fish are referred to as primary consumer, and the majority of primary consumer are herbivore. Ew! What exactly is this? This is the Cicadidae family. Alternatively, very young. Cicadas are herbivores, which means they eat mostly plants. The young nymphs use their modified mouthparts to feed on plant root juice and saps, whereas molting cicadas eat twigs. Mushrooms are herbivores because they are fungi. They have their own distinct way of obtaining nutrients from soil and other substrates. They decompose organic matter to obtain the carbon they require. Then there's carnivory. Carnivory occurs when a predator consumes meat rather than plant and as a result kills its prey. Carnivorous organisms are those that consume meat. We discovered a spider species there. The majority of species are carnivore, either trapping or hunting flies and other insects in their webs. Spiders can swallow their prey whole, so they inject it with digestive fluid and suck out the liquid-filled remains. Finally, there is parasitism. The propagation of parasite is generally passive. From the outside, they hunt, kill, and feed on their prey. Invaders living cells consume nutrition and other host benefits, and causes harm to the host. A mosquito is a parasite that feeds on human while transmitting the disease malaria, lies on human and cows, ectoparasite, and tapeworms in a cow and human intestines, endoparasite. We discovered 4 types of prey defenses interaction: chemical, mimicry, cryptic correlation, and flashing correlation. First, there are chemical prey defenses. Trimeresurus wagleri, also known as Ulacapatoko, is the species. Snakes use venom to avoid predator or to catch prey. The venom gland of the snake are located on the both sides of its cheek. This snake's venom is a blood poison, hemotoxin, and a cell poison, cytolytic, both of which act on blood vessel system and tissues, causing bleeding in the victim, either inside or outside the body. Following that, millipedes may secrete slimy substance mucus through their body. Then there's mimicry. Mimicry is an interesting subject, especially when viewed in terms of selective forces and evolutionary strategies. Mimicry is a system that has evolved independently many times in plant and animals and consists of a signaler, a signal receiver, and a model. Despite having a different body plan than ants, at least 13 spider families share an ant-like resemblance. Quantifying the degree to which spiders resemble model ant in shape, size, and behavior will help us understand the evolutionary pressure that have facilitated mimicromorphy. Cryptic correlation. Color concealment or color protection allow prey to flee predators. It employs pattern shapes, posture, movement, and behavior to obscure prey. Fish, reptiles, and ground nesting birds are all examples. For example, can you spot the differences in this image? I give you about 5 seconds to look carefully. This is only a simple level, okay? Can you spot it? Yeah, it's here. It is cicadas. But poor little guy, this cicadas almost died when we found it because it has difficulty shedding its shells. Most are the insect skin that cicadas shed or leave behind when they become adults. Explained by Nancy Troiano, PhD, a board certificate entomologist. Okay, now let's proceed to the hard level. Begin in 5 seconds. Take a close look, okay? Yes, it is there. Blanchard cricket frog it is. The last prey defenses in interaction is flashing correlation. Trimeresurus wagleri has yet another characteristic. The temple egg snakes, like most other venomous animals, have a bright body color that makes it easier to identify. Egg-temple snakes have large heads and are green, turquoise, or black with yellow, light green strips, and black spots as adults. Some adult male snakes, as well as some young snakes, are bright green. So that's all we can say about our field trip BIO464 process in ecology. We hope that this type of activity will continue for our junior in the coming semester.